Well, look,
Right.
okay. So welcome everybody to the second WHS Journal Club. I'm Dr. Mark Weatherall, I'm supposed to be the moderator of the Journal Club, and I to find myself today as also the presenter. So I would very much encourage anybody that is here or watches a recording of this Journal Club today to step forward and volunteer to present papers. If you have a paper you want to present, you're very welcome to do so. We're very interested to talk about anything that relates to headache. If you would like to present but you don't know what to present, then please come to us, and there are plenty of very good papers that would be very good to discuss. So today I'm going to talk about a, a piece of basic science which I think is probably the most important um, headache paper or basic science headache paper that's been published in the last few years. Um, so I should now be sharing my screen, and I hope you can all see it.
Yes.
Great. Okay. So this is a paper that was published in the journal Science last July. And for those of you who are not intimately familiar with the sort of the kind of scientific journal world, Science is one of the probably top three or four scientific journals in the world. I think, you talk about Nature, you talk about Cell, you talk about Science. So to have papers published in this journal is quite something and quite rare. And as we'll see, this was quite an impressive piece of work. So obviously, I don't need to tell you all that migraine is the by far the commonest debilitating primary headache disorder, and that migraine attacks can have multiple phases, and it's not clear how these interact. This is a picture of a British clinician called Nat Blore, who worked at the National Hospital and the City of London Migraine Clinic in the 60s, 70s, 80s, and 90s. And I was privileged enough to meet Nat about 15 years ago, shortly before he died. Nat was one of the people who sort of first really pointed out this clinical description of migraine as a condition that could have phases that uh, extended just beyond the headache. And this is a diagram from a paper that he published in the 1980s showing the prodromal phase, the aura, the headache phase, the importance of sleep resolution, which is a process we still really don't understand at all, and then the postdromal recovery phase. And obviously, as I say, it's not clear really how these phases interact, because it's perfectly possible, in fact, for people to have some of these completely individually. We've all seen patients who have just migraine aura and then don't really have any other manifestations of a migraine attack. And of course, we've seen many more patients who just have the migraine headache phase. So, how do these things sort of, these come together? That's not really very well understood. So, these are pictures on this slide. This is a picture on the left-hand side from Peter Goadsby's Nature Review article of a few years ago that just sort of outlines, some of the connections and circuitry that's active in a migraine attack, with the idea that the migraine headache phase, at least, is driven by activity within the trigeminocervical complex in the brainstem, and that there is this sort of feedback loop via the trigeminal ganglion, which is going to be quite important in today's story, out to the extracranial vasculature, predominantly dural and meningeal blood vessels, with the release of vasoactive neurosubstances around the blood vessels and within the blood vessels, so obviously particularly CGRP, calcitonin gene-related peptide, and then a sort of feedback loop back to the brain, and then activity sort of extending through brain levels until it hits the cortex and our consciousness, and we become aware that we're having an attack. And also, this provides quite a nice explanation for the potential relevance of painful or abnormal sensory input from the neck and from other parts of the trigeminal system. However, what it does not explain is how aura processes interact with that, because of course aura is, as we understand it, the experience that is the experiential substrate of a physiological process called cortical spreading depression, which is a slowly spreading wave of overactivity and underactivity that has a predilection in patients with migraine for the occipital cortex, thus causing visual aura, and is associated with and causes spreading oligemia. So the upper picture on the right hand side is an ECG recording showing a spreading wave of cortical silence followed by a resolution a few minutes later, and the bottom right hand picture is the famous first demonstration by PET scanning from Martin Lawrence and Jens Olsen from the 1980s, showing spreading oligemia quite clearly crossing the boundaries of the kind of major intracranial blood vessels. So that's the sort of background. How does aura cause migraine headache? Does it cause migraine headache? And the authors of this paper start off by saying quite correctly, I think, that it is not currently understood how pathological events in the cortex, in other words, CSD, can affect peripheral sensory neurons. And by peripheral sensory neurons, they are predominantly interested in the link between what is going on in the cortex and what is going on in the trigeminal ganglion, because the trigeminal ganglion, which is an important relay station in this feedback loop between the central trigeminal cervical complex and the peripheral blood vessels, is or has been regarded as sitting outside the blood-brain barrier, so being peripheral. And we know that the medications that we give for migraine, up to and including the triptans, do not, under normal circumstances, cross the blood-brain barrier. And it's pretty clear that even under abnormal circumstances during migraine attacks, that they don't cross the blood-brain barrier either. So this is, again, this raises the question, how can a peripheral input then have an effect on central processes? But also then, how can something that's going on within the brain, cortical spreading depression, affect these external systems? So the objective of the paper was to identify a communication pathway between the central and peripheral nervous system that might explain the relationship between migrainous aura and headache. So how did they do it, and what results did they get? Now, I've basically sort of put together the methods and results because the paper basically provides a series of 7 individual sections that the authors provide a sort of stepwise series of evidence for their proposed communication pathway. So it's quite complicated to sort of go through all the methods and then go back and do all the results. So I'm doing them one section at a time. So the first thing that they showed was that CSF has got direct access to the trigeminal ganglion. And of course, this, again, goes against the idea that the trigeminal ganglion is actually on the far side of the blood-brain barrier. Now, in order to demonstrate this, they used a technique called gradient index lens, which is basically allows a direct view of the trigeminal ganglion in vivo. And what they used was a transgenic mouse model, in which mice who were exposed to TRPV1 agonists, such as capsaicin, would express a calcium-sensitive fluorophore. So basically, in the sensory neurons in these mice, If the system was activated, then they would express a fluorescent material that would allow direct observation of activity. So what they could do with this is that they could look at CSF inflow, but they could also look at sensory nerve activity at the same time. So the first step here was that they injected capsaicin into the CSF. And what happened was that it appeared within the trigeminal ganglion within a few minutes of being injected into the CSF, and it drove an increase in calcium activity. So the graphs that I've reproduced here basically show the right-hand side. This is the presence of activity, tracer activity within the CSF. And then on the left-hand side, this is the consequent activity. This is cyclic GMP. So this is calcium-related activity within those neurons. Following the injection of capsaicin, the level of activity went up in consort with the amount of CSF inflow into that area. So that was the first thing that they showed. If that CSF would access the trigeminal ganglion and could potentially impact upon activity within the trigeminal ganglion. And then as part of this, they also then said, well, okay, is it possible for a process in the cortex to do that as well? And what they used was an adenovirus transfected cell line in the dorsal cortex, which would express a fluorescent protein. So again, they injected this, and the cells started to express the fluorescent protein, which then was seen in the trigeminal ganglion. So this then kind of provides a first sort of clue that actually the trigeminal ganglion is not purely a peripheral structure, but it has a central portion. So the second step was for them to use an MRI tracer. And so what they did is that they first kind of looked at sort of detailed MRI imaging in anesthetized mice. So that showed that the trigeminal ganglion was located close to the basal cisterns and basal cerebral arteries. And of course, we know that in humans, the trigeminal ganglion sits within Meckel's cave, which is basically a kind of CSF reservoir. And what they did is they injected a contrast agent, gadolinium, into the CSF. And that appeared almost immediately within the trigeminal ganglion. And notably, the most of it was within the proximal portion of the trigeminal ganglion. So what they then hypothesized was that, unlike most nerves, as they leave the CSF, there is a perineural sheath, and that has tight junctions that stops the efflux of CSF along the perineural sheath at some point. They hypothesized that that was not the case for the trigeminal ganglion route, and they demonstrated by immunohistochemistry that actually the first third of the proximal third of the trigeminal ganglion is actually CNS, it's central nervous system. So they used, this is number 3, they used astrocytic aquaporin 4 staining. What you're getting is immunohistochemical staining of astrocytes, so central nervous system cells. And as you can see, there's a very clear border proximally within the CNS that's staining all the way, and a third of the way into the trigeminal ganglion, that staining stops. And everything beyond there is peripheral, so there are no astrocytes, no aquaporin 4 expressed. And they were able to demonstrate that essentially what happens is that as you can see from the top right picture, you've got a central nervous system portion of the trigeminal ganglion, a peripheral nervous system portion of the trigeminal ganglion. The dura and the arachnoid, the tight junctions exist, but only distal. So proximally, it's possible for CSF to reach the proximal portion of the trigeminal ganglion and be absorbed and affect processes at that point. And then once you go beyond those tight junctions, any drainage from the trigeminal ganglion actually goes outwards towards the cervical lymphatics and not back inwards. So, having demonstrated that part of the trigeminal ganglion sits within the CSF, within the central nervous system, and that CSF can access it, the next question is, well, okay, if CSF accesses it, that's fine. But if something happens, further away in the brain, so, in this case, the visual cortex, because that's where most migraine aura occurs, can that still have an effect, basically at the other end of the brain, in and around the trigeminal ganglion? So in this case, what they did is they injected gadobutyl into the visual cortex. And what they found was that it indeed did drain down towards the basal cisterns and ended up in the area of the trigeminal ganglia. So this is the top picture here. This is just a picture of them doing this, injecting contrast agents into the visual cortex. Now, some of the other areas that have been postulated as potential areas where there could be this link between the central processes and peripheral processes, so the specifically things like the meningeal lymphatics and the parasagittal dura, they compared how much of the gadobutyl ended up around the trigeminal ganglion in the basal cisterns and how much ended up in the sort of parasagittal dura. And they found that about 4 times as much ended up around the trigeminal ganglion as around the parasagittal dural spaces. And interestingly, there was a very strong ipsilateral concentration. So the contrast agent that was injected in the visual cortex largely ended up sitting in the basal systems around the ipsilateral trigeminal ganglion. So obviously, if you're postulating some form of process that links migraine aura and then a unilateral headache response, and many of our patients, not all of them, but many of our patients will describe one-sided aura, maybe aura in the left visual field, and then a contralateral headache, right-sided headache. This is very interesting because this potentially provides a potential kind of mechanism by which something that's happening in one visual cortex could then stimulate what's happening in the ipsilateral trigeminal ganglion, and that would certainly potentially at least result in one-sided visual aura symptoms and headache on the other side. So that was quite a nice demonstration. And then what they then went on to show is that, okay, so fine, visual cortex, something for the visual cortex ends up near the trigeminal ganglion. That's good. What about what actually is happening during migraine with aura? So in this case, they use again a mouse model, THY1 CR2 mice, which can have CSD induced optogenetically and also by the infusion of potassium chloride. So initially they induced CSD after the injection of a CSF tracer, and the idea is that CSD is a metabolically active process, and so what will happen is that the CSF tracer will be taken up within cells, but then washed out. And so again, what they found was that when you did that, there was a 4-fold increase in tracer influx as a consequence of CSD, and that lasted about 5 minutes, and much higher on the side where CSD had been activated than on the other side. If you then kept on inducing corticosplenic depression, so mimicking a longer period of aura, over an hour, that would cause the tracer signal to fall away. This is the top right-hand picture. So essentially what's happening is that's the tracer is getting taken up quickly, but then is being washed out of the area where there is CSD much more quickly and much more actively, rather than by comparison with the controls and the contralateral side. So again, what's been shown is that CSF uptake occurs during CSD, and there is active washout of extracellular solutes from cells that have been undergoing CSD. So then the question becomes, well, what are those extracellular solutes? And one of the answers to that is, well, some of them at least are potentially relevant inflammatory CSF proteins. So then this is again a kind of another but related piece of work that they've done. So they've used a sort of bottom-up mass spectrometry, and essentially this is a technique that kind of identifies potential proteins and from genes that are found within the CSF. So they found a kind of CSF proteome of about 1,400 genes. Of those, about 150 of them changed in amount after corticosteroid depression, and about 20 of them were kind of significantly upregulated. Now, they then kind of went back and looked at, well, okay, there's a lot of genes, there's a lot of genes and a lot of potential proteins that are in the CSF and maybe change when CSD processes happen. How many of these might be ligands to known or postulated trigeminal ganglion receptors? So the answer to that question was about 60 of them potentially could be ligands to proteins that are thought to be receptors at the trigeminal ganglion. And nearly all of those, if you look at them functionally, and that people, they're doing a kind of making an assumption of which ones may be functional, which ones maybe not. But assuming the ones that are, most of them seem to actually have a stimulatory activity. And of those, if you then look at cross-reference those two pieces of work and say, okay, of the ones that we've seen to be upregulated after CSD, that potentially could act at trigeminal ganglion receptors, how many did they identify? Well, they identified 12, and these are the ones in the diagram. This is sort of what's called a volcano plot of activation of or amounts of the proteins before and after CSD. And the ones that have got the black circles around them are the ones that are significantly upregulated. And one of those genes is the CALCA gene, which is the one that encodes for CGRP. And there are a number of others. Many of these have been, the protein products of these genes have been shown to be potentially involved in hyperalgesia inflammation. The gene that encodes PACAP is also in there, upregulated, but not at such a significant level. And then if you look at the receptors that they're looking at, these trigeminal ganglion receptors that could be paired with these, yes, they're found on the trigeminal ganglion, but they're also found more extensively elsewhere within the CNS as well. So finally, trying to put this all together, can changes in what you see in the CSF after CSD actually induce activity in trigeminal sensory neurons? So we've gone back to the initial technique of literally looking at what's happening with in trigeminal ganglia in mice in vivo. And what they did is that they basically directly exposed them to CSF that had been collected. Well, they looked at what was happening in this scenario where they induced CSD, and that showed a five-fold increase in activity in the trigeminal ganglion neurons with a latency of about 11 minutes. They then took the CSF itself and exposed naive trigeminal ganglion to the CSF to demonstrate that this increase in activity was likely to be mediated through the CSF and wasn't something that was just a more direct link. And that also increased activity. But then what they did is that they then went back and they collected more CSF after about 2.5 hours, and that did not have an effect. So the conclusion was that the increased activity in the trigeminal ganglion was a consequence of early changes in the CSF constituents, presumably related to extracellular washout as a consequence of CSD. They also showed that if you directly applied CGRP alone to the trigeminal ganglia, that also it caused an increase in activity, which provides circumstantial evidence that CGRP may be one of the proteins that's involved at this point. So it's quite a lot of detail to go through. Essentially, the conclusion was that they demonstrated, and I think pretty convincingly, that CSF can at least transport solutes from the cortex to the trigeminal ganglion. And this provides a non-synaptic route of communication between central nervous system and peripheral nervous system areas. And I think, very interestingly, this cortex to ganglion, the speed of the flow of information and the laterality is consistent with observed, or at least potentially consistent with observed migraine phenomena, which is very interesting. CSF composition quite quickly normalizes, and they concluded, well, other processes must drive headache at later phases, and they speculate that this could be an intrinsic effect of CGRP within the trigeminal ganglion. There is also the question of where else CSF goes, and of course, it is known that CSF can affect the dura mater via bridging veins into the meningeal lymphatics and the parasagittal spaces. And of course, this is one of the things they looked at, and they said, well, we do find that if you inject contrast medium into the visual cortex, it does end up in these other spaces, but not nearly as much or nearly as quickly as it ends up around the trigeminal ganglion. And they conclude by pointing out that, while CGRP is there, and we know that CGRP is important in migraine processes, there are other protein receptor pairings within this sort of CSD upregulated proteome, and they suggest that these might be potentially new pharmacological targets for migraine. So what do we make of it? I mean, what to make of this? Well, look, the first thing is this is the first migraine-related publication in Science for more than 40 years, which is an achievement in itself. The authors, it's a really high quality kind of multidisciplinary team. About 50% of the authors are from the University of Copenhagen. For those that you don't know, Copenhagen is kind of one of the big centers of headache research in Europe. The Glostrup Hospital there, this is where a lot of the work has been done on functional imaging in migraine, CGRP, and so on. And then there's a big sort of collaboration here with teams in America at Rochester, at the National Institute for Neurological Diseases and Stroke in Bethesda in Maryland. There's the involvement of a biotech company in Cambridge, Massachusetts, and also of the Max Planck Institute of Biochemistry in Germany. The techniques that they've used are, I think, very kind of well validated. The immunohistochemistry is a long-standing technique. The optogenetic techniques and the way that they're able to visualize, in vivo activity by using these sort of transgenic fluorescent producing mice is I think again really quite well validated across the board in terms of trying to understand the expression of activity within brain cells across a variety of different processes. And the findings do potentially accord well with the clinical features of migraine in humans. But what it doesn't really explain is how, so it gives us, it's quite convincing for, yes, there is a potential way that processes that occur in CSD could then create activation of the trigeminal ganglion. But how then does that really kind of cause the experience of headache? Rami Bernstein from Harvard, who's very sort of well-regarded migraine researcher, pointed out that's all it's great, it's a really interesting study, but why doesn't stimulating the trigeminal ganglion in this way cause pain in other areas innervated by the trigeminal nerve, in the face and the teeth? I mean, obviously we know some migraine patients do experience that, but most migraine patients get pain in kind of V1 and not much else elsewhere. And then, what are the sort of broader implications of this study for, for other, headache types? We know that there were CSF changes, for example, in post-traumatic headache. What are the impact of sleep disruption? What's the whole sort of, impact of, the kind of washout of extracellular solutes from cells? Where does that take us? And and I think that's one of the exciting things about this whole, this paper is that it kind of opens up a kind of completely different area of interest for us in terms of, the science of migraine. So really, just the sort of takeaway that I got is, look, I think the authors successfully identified a communication pathway between central and peripheral nervous system that might explain the relationship between migraines, aura, and headache. And if the findings are relevant to human migraine physiology, then it might lead to the discovery of new pharmacological targets for migraine treatment. If you are interested in this whole area, then there is one other, I think, really important publication that came out last year in the Journal of Clinical Investigation, which actually was about the meningeal lymphatic CGRP signaling. So this is the kind of other potential sort of conduit between sort of cortical processes and if you like, the kind of headache kind of feedback loop that's active. And so this is a very nice paper that was published in JCI last year, and obviously we'll circulate the details, and maybe somebody would like to talk about it at one of our other meetings. So there you go. I'm going to stop sharing at that point and see if anybody has any comments or questions.
That was a very brilliant presentation, Mark. Really made that very complex paper, how you have summarized it so beautifully that I could understand many of the things which I couldn't understand in the first reading. Do we have any questions from those who are here online? If not, I may start a few questions of my own. Professor Momozo.
Yes, thank you for your how to think and how to explore the solutions. And one I would like to clear is how frequent CST started. It means CST. How frequently occur CST?
Well, so I mean, obviously, in the studies, what they did. I mean, I think there's some interesting things there. So they induced CSD in two different ways. So they induced it optogenetically, so using kind of light sources in mice that I think are transgenically prone to CSD. So they're basically, it allows them to have CSD induced more easily. Now, in a sense, that's a kind of migraine model, because we know that all human brains potentially can undergo CSD, although actually in a lot of cases CSD is a pathological process. After subarachnoid hemorrhage or stroke or something like, or those things like that. So one of the interesting things about human migraine brains, if people get aura, is the fact that they can undergo CSD without it causing damage in the vast majority of cases. So that's how they induced it first time. But then when they were trying to induce it repeatedly to kind of mimic, if you like, the kind of metabolic stress of a longer migraine aura in humans, they actually used potassium chloride, and they basically just it's sort of kind of an irritant to the brain, and that will set off CSD in models. So they did that for up to an hour in order to kind of mimic the sort of longer period of kind of the metabolic sort of after effects of CSD. But you see, one of the questions, I mean, there's a few small things. So, for example, when they induce CSD, they induced it in the frontal cortex of the mouse, which is not necessarily the same as the occipital cortex of the human being. So, obviously, one of the kind of fundamental questions about this paper is what they're seeing in their mouse models, actually would that actually transmit to what is really happening in human beings? Um, and we don't know the answer to that question, but there's a whole series of interesting kind of hypotheses, and you could envisage potentially the possibility of doing some interesting human studies if you could persuade people to have CSF sampling in the context of migraine attacks, whether you might be able to demonstrate some similarities in, changes between the kind of resting CSF proteome and, an in migraine, and in, human in vivo migraine CSF proteome, and see if you could identify in humans the same sort of changes that they see in the mouse models. And if you could do that, which is not actually something that would be necessarily that difficult to do, then that would be a very interesting that would be a very interesting demonstration if you could demonstrate a similar sort of pattern of changes.
Thank you.
I mean, obviously, it doesn't. Again, it also doesn't answer the question of, well, I mean, how does migraine, what initiates migraine in people that don't have aura? And because again, that's a long-standing question, isn't it? Do people who apparently don't have aura, do they have silent aura, or do they just not have aura processes at all? That's number one. And number two, it doesn't answer the question of why some people get aura but they just don't get, headaches afterwards. Although that's probably more easily explained on the basis of, well, presumably different people's aura process will, end up, causing different levels of extracellular solutes and CGRP and so on. And there may be different responses to that over the course of a lifetime. So, like all good studies, it raises as many questions as it answers.
Yes.
Absolutely. Yeah, so I'm just a few comments. Well, if you ask what's the fundamental question that's being answered in this paper, it came out clearly after 40 years. A lot of hard work has gone into it, but being the critic that I am generally, and if, I know you have done a critique as well. A few things I think you have just kind of, in response to the question put forward by Momozo, you have really thought the same, and I'm sure that many of us do think along the same lines about this mouse model. Firstly, I mean, it's one of the good models that we have here to study the disease. Do we have better models? Well, we can debate on that. But given the situation, given the mouse model that we have, what we do come across in this study are there are a couple of things that is notable. That once there's an injection into the CSF compartment, there's this increased calcium activity. Now, do we try to see whether the injection itself has induced this activity? What if there was no injection? It's just difficult to know because you are possibly injecting. There is possibly a nociceptive input there when it comes to injecting that. Just a wild thought. Perhaps they have taken care of it. But even if it is injected, and let's say the question that's answered is that there is a, a connection between the center and the periphery. Now, logically, in everything in neurology, we understand that yes, there has to be. With many pain disorders where there is a central sensitization or a central disorders of pain, there is a peripheral pain, but there is a central etiology. So theoretically, it's there. I mean, there is something happening in the center and then something's manifesting in the periphery. Now you could argue that, well, now it's theoretically it's there, retrospectively you give medications, drugs which act in the CNS is working in reduction of pains and migraines. Now, you could say that, well, this is what you've demonstrated. But, it's interesting to also look from the other point, the school who would like to be excited about trying to prove the other way, whether there's a bidirectional relationship in the sense, what would happen if something is changed in the periphery and whether it's demonstrating changes in the center, in which case this is a lovely model of bidirectional relationship because it clearly we know an external compression headache can produce a migraine. So there's something starting from some point, or an entrapment syndrome producing, zygomaticofacial compression producing a migraine. Now that's something which perhaps is going to be the next stage for this kind of studies. And the other thing about is CSD. I mean, you have said it already. It's not just in migraines; it's there in seizures, it's there in stroke. With all of these, and the lack of clear demonstrations. And of course, the mouse models are optogenetically or otherwise, you're inducing a CST, whether how much of that is spontaneous, and that too in migraines with aura, and that too visual aura. I mean, there are other auras, and there's migraines without aura. So the big question is again, are we looking at another disease? Migraine with aura or migraine with visual aura is a very specific entity, which is just a very small segment compared to all of the other migraines which people experience. And I think I think you're trying to answer, but this is my last question, just rather on just observations and also about the ipsilateral trigeminal activation. I mean, it explains the ipsilateral pain. But as you rightly observed, why contralateral? So these are the
Well, I
questions.
think no, but I think that is if you presume you've got a right. I think what they're saying is if you've got, for example, right-sided aura, so you've got right visual aura, you're going to get left-sided symptoms with your aura symptoms, but then you're going to get ipsilateral, you're going to get right-sided trigeminal activation, so you get a right-sided headache, which is, I mean, it's quite often which is quite often what one sees. You do get patients that say, "Oh, well, I get, the aura's over here and then the headache's on the other side," which has always been actually a really interesting observation, isn't it? So that would actually fit, but then it wouldn't explain, if you then people get, more generalized headaches or so on. I mean, I think the point that you make about the kind of the bidirectional nature of communications between sort of central and peripheral processes. I mean, I think that's absolutely central. I think this is happening a lot in migraine processes, and we make use of that every time we do an occipital nerve block for somebody with migraine. We're changing a peripheral input into that central system. I mean, I personally think probably that's how Botox works as well. Again, it's modulating the way that all these sensory neurons send information back into the brain.
Or even a
So,
simple massage.
yeah, yeah. Well, no, exactly. For people that, I mean, people who've got more hair than me that kind of pull their hair tight when they have a migraine, then it's helpful. I think Momo could do that. I can't. So I think, look, I mean, clinically, from what we observe and what we do in migraine, we are taking, advantage of the fact that migraine, there is, yes, we know that there are central processes active in migraine. That migraine biology, that diathesis, that tendency to get migraine, the genetic basis of it, probably resides predominantly in a sort of brain propensity to be headachy and the activation of a whole series of networks within the brain. But the things that feed into that, there is a lot of peripheral input into that, and I think that's that it definitely going both ways. I think I mean, my personal view on kind of where migraine with all sits. I think Elizabeth Loader wrote a really nice paper a couple of decades ago, sort of about the idea that migraine is almost a kind of modular kind of disease, that there are lots of different things that can happen, and there is often an association of them, but they can all happen kind of individually. And that probably ties in with what we start to understand about kind of migraine genomics. Different bits, different, sets of genes in different areas of the genome seem to be associated with, for example, the tendency to throbbing headaches, the tendency to headaches with nausea and vomiting, an aura and so on. So there are probably a number of kind of, migraine modules that that often sit, can sit separately, but often sit together. And I think, again, this comes back to the question of how all of these things kind of link in together. But it does, and it creates the heterogeneous picture that we see. And that's what makes it interesting, doesn't it? Because it's what I think there's lots of interesting things there. I think migraine is, for the questions, lots of questions that arise from that. Why is migraine much more heterogeneous than, for example, cluster headache, which is much more sort of tight. There's a sort of tight biology to cluster headache that is very, very similar, whereas migraine is a much more disparate, heterogeneous condition. Anyway, lots of sort of questions to that we can discuss again at at length. I mean, I come back to this. I think the value of this is, I think it's very rare to come across a paper that makes you think, or kind of opens up a kind of completely kind of new kind of set of kind of thoughts about how things might work. And there is a literature on the behind this where people have become increasingly interested in what's happening with CSF in migraine. I think there's an increasing literature on the role of the lymphatic system in migraine. I think there's a lot of kind of this is a coming area in neurology generally. I think, isn't it? There's a whole load of stuff that's in the brain that people haven't really been kind of that interested in, and suddenly everybody's discovering the fact that there is more to the brain than just glia.
Yeah.
So and I think that for all of these things, but I think this, as I say, I think that the success of this paper is that it kind of makes us just kind of think and wonder about what we're
Yeah.
looking at in a different way.
Absolutely, you've answered brilliantly. Yes, I mean, I would be very curious to know from the authors whether they have looked at the contralateral trigeminal ganglion. With ipsilateral, they did find activity, and if it's mediated through the CSF, we would
Mm.
probably expect something to spill over to the other
Yeah.
side.
I think the answer is, so yes, the answer to that question is yes. There is, I think there is activity in the contralateral trigeminal ganglion, just not as much. Um,
Sure.
and I think my understanding was that activity was probably higher than just what they would sort of control. And the other thing that you mentioned, actually, that I didn't mention specifically was not quite an answer to your question, but you're talking about experimental induction of CSD, and and does that then have a kind of intrinsic nociceptive effect. One thing they did look at was about the impact of potassium levels in the CSF itself,
Oh,
when they use potassium to induce and to maintain CSD. And
yes.
they didn't, and they specifically looked at that, and they didn't find a difference between potassium levels in CSD, induced CSF and non-CSD. So I think that was one specific thing that they kind of looked at and ruled out. But yes, again, all of these models, sit on they all teeter on that, that fulcrum of, is what we're looking at experimentally. How relevant is that to what actually happens in human brains when we actually have migraines?
And the other exciting part, which I just thought about, is about the unilateral symptoms in patients where the pains don't just sit there, but would affect the limbs as well. So if it's a CSF
Mm,
pathway, then we'll have all of these mediators going all the way down the
mm,
spinal cord, the
mm,
sensory roots.
mm,
So stretching this paper a bit, possibly can even think about why people experience hemi-body pains with migraines,
mm,
the
mm.
same side of the headache.
Yeah.
Not everyone, many of them.
No, no, but yeah, it's again, it's an interesting observation, and not that easy to explain with what we understand thus far.
Yeah. Yeah, as you said, the concentration kind of goes lower as you go to the opposite side and perhaps even goes lower down than your axis.
Mm.
So bulk of the symptoms are around the head and neck. Lovely. Thank you so much. Thanks. That's great. And thank you. And I think
So,
we are just getting close because you've got, I know you're busy with other
well,
things.
again,
Yeah.
all I wanted to say is, again, thank you very much for everybody that's attended, here and now, and to any of you that are watching this, recording later on. I hope you've enjoyed it and found it interesting. And again, I would just finish off by encouraging, people to just to step forward and present. I think we've got a series of dates. They're mostly on Monday lunchtimes. It's just it's kind of it's good experience and actually kind of fun just to take a paper and just to kind of dissect it. And I think we're going to be a reasonably friendly audience to do that in front of. So really good to have one or two volunteers to do the next couple of months. And as I say, if you don't have a paper, we can give you a paper to do, or we can suggest a paper to do. But if you have something you want to present, that would be great.
Thank you so much. Thanks a lot.